We breathe it constantly, right? Oxygen, it feels so fundamental, so life-giving.
Absolutely essential, yeah.
But what if I told you that same gas, the one keeping us alive, could actually be dangerous,
Yeah.
toxic even?
It sounds counterintuitive, but that's exactly what we're diving into today, the whole paradox of oxygen toxicity.
Like how something so vital can, under certain conditions, turn harmful.
Precisely. And we're not just guessing here. We've got some really solid scientific grounding, a whole chapter actually dedicated to oxygen toxicity.
All right, so think of this as getting you up to speed on a pretty complex but really important bit of science. Our goal today, to get beyond just oxygen good and understand the risks when things aren't, let's say, perfectly balanced.
Exactly. It's a story that actually goes back centuries, from people having early suspicions to, the nitty-gritty molecular details we understand now, and what it means for things like medical treatments. So where do we kick things off?
Let's go way back to the beginning, almost. Joseph Priestley, the guy who, gets credit for isolating oxygen. He apparently had some early thoughts about this.
He really did. It's quite insightful, actually. Priestley speculated that his dephlogisticated air, what we now call oxygen, might be good for illness, but maybe not so beneficial for healthy folks.
Oh.
Yeah. He used this analogy of a candle burning out way faster in pure oxygen. And he wondered if living in that kind of air might make us, and this is his phrase, lit out too fast, like we'd burn through our animal powers too quickly.
Wow. That's pretty forward-thinking for the 1770s, almost like he sensed there was a limit.
It's a fascinating early hint at hyperoxia's potential downside, yeah.
Okay, fast forward about a century, Paul Burt enters the picture, and things start getting more scientific.
Absolutely. Paul Burt, late 19th century, his work was, groundbreaking. He was experimenting with oxygen under high pressure, hyperbaric oxygen.
Right.
And he very carefully documented its toxic effects, especially on the brain, the central nervous system. He found that high-pressure oxygen could cause seizures. That was huge.
And that's the Paul Burt effect?
That's the one. We still call CNS toxicity under pressure the Paul Burt effect.
Okay, so Burt nailed the brain issue under high pressure. But the source material says he kind of missed something else.
He did, yeah. His focus was really on the high-pressure side. He didn't really investigate the effects of just breathing high concentrations of oxygen, like 100%, but at normal atmospheric pressure, particularly on the lungs.
Oh, okay.
That piece came a bit later, thanks to Lorraine Smith. She showed that you could get pulmonary oxygen toxicity, lung damage, even without the high pressure.
And that's the Lorraine Smith effect, I assume.
You got it. So these two, Burt and Smith, they really laid the foundation. Burt, CNS, high pressure, Smith. Lungs, normal pressure, high concentration. It showed early on it's not just about enough oxygen. Too much in the wrong way targets specific systems.
And the story keeps going. There's a John Bean effect mentioned, too. Sounds kind of serious.
It is, unfortunately. John Bean's work, mid-20th century, showed that if you push hyperbaric oxygen exposure even further, beyond just seizures, you could get irreversible neurological damage, even death.
Okay, that's sobering. So all this early work started painting a picture of, like, safe limits.
Exactly. They started figuring out roughly how much humans could tolerate. The early thinking for healthy adults was maybe about 3 hours at 3 atmospheres absolute. That's like being 20 meters underwater pressure-wise. Or only about 30 to 40 minutes if you went up to 4 atmospheres absolute.
And they noticed different effects at different pressures?
Yeah, that was interesting. Around 3 ATA, it seemed to primarily hit the nervous system. But even down at 2 ATA, the lungs could be affected independently.
Okay, so we have the history, the key players, some early safety ideas. But what's actually happening inside us at the molecular level? Why does oxygen become toxic?
Right. That takes us to the free radical theory. It's the leading explanation. The basic idea is when you breathe higher than normal levels of oxygen, your body starts producing way more of these things called reactive oxygen species, or ROS.
Free radicals, I've heard of those. They're unstable molecules, right? Cause damage.
That's the idea. Think of them as overly reactive molecules looking to steal an electron from somewhere else to become stable. And oxygen itself, its molecular structure, makes it kind of prone to forming them.
How so?
Well, inside our cells, oxygen can easily pick up single electrons in reactions. This forms something called a superoxide anion. That's a key free radical. And it doesn't stop there. That superoxide can then lead to other reactive things like hydrogen peroxide and these really nasty hydroxyl radicals.
And these radicals, what kind of damage are we talking about inside the cell?
They can mess things up pretty badly. They can oxidize parts of enzymes, sort of switching them off. They can interact with DNA, which is obviously not good. And they can cause something called lipid peroxidation. Basically, attacking and damaging the fats that make up our cell membranes disrupts the whole structure and function.
Okay. Our source mentions a specific hypothesis from Bovers and Chance. What was their take on it?
Yeah, Bovers and Chance, back in the late 70s, proposed a really influential idea focusing on the mitochondria.
The cell power plants.
Exactly. They suggested that high oxygen levels make these mitochondria churn out a lot more hydrogen peroxide. Specifically, those superoxide radicals generated inside the mitochondria are the main source.
Okay.
And this extra hydrogen peroxide then kicks off other problems, that lipid peroxidation we mentioned, enzymes getting inhibited, the cell's antioxidant defenses getting overwhelmed. Ultimately, it messes with the mitochondria's ability to produce energy. It's like a cascade failure starting in the power plant.
That makes sense, like a domino effect. But the source also adds a little caution about the human evidence.
That's a really important point, yeah. Animal studies are pretty clear. You see ROS go up before seizures happen with hyperbaric oxygen. But in humans, it's, well, a bit more complex.
How so?
Some studies do show signs of increased free radical activity in people exposed to HBO, but often those signs seem to return to normal pretty quickly afterwards. So while the free radical theory is definitely the front runner, proving it definitively in every human situation is still, ongoing work.
Right. It's rarely simple. Okay, so we have the molecular suspects. What does this damage actually look like? What's the pathology?
Well, the actual physical changes have been documented quite extensively. Ballantyne's work is mentioned specifically. The key thing that determined what gets hit the brain or the lungs is a combination of the oxygen's partial pressure.
So how concentrated it is and the pressure it's under.
Exactly. And crucially, how long you're exposed. Those two factors, pressure and duration, are critical.
But the good news is, early on, the damage can be fixed.
Yes. Thankfully. In the initial stages, the effects are generally reversible if you stop the high oxygen exposure. But if it goes on longer, recovery takes more time, and eventually, yeah, you can get irreversible damage. Though the really extreme stuff seen in some animal experiments isn't typical of clinical HBO.
The chapter mentions effects on pyruvate and lipids, too. Why is that significant?
Right. That's interesting because it shifts the focus a bit. Initially, people might have thought oxygen toxicity was mainly about disrupting how we use sugar for energy. But these findings, changes in how pyruvate and lactate balance out, and less fat getting incorporated into tissues, they point more towards those damaging lipoperoxides, the breakdown of fats being a really central part of the injury process.
Got it. It sounds like scientists then tried to figure out how to measure or predict this toxicity risk. Something about power expressions from Israel.
Yeah, researchers at the Israel Naval Medical Institute did develop some mathematical formulas trying to quantify the risk. For the lungs, they had an equation looking at how much vital capacity lung function might decrease based on oxygen pressure and time, and another one for how quickly it might recover. They did similar things for CNS toxicity, trying to calculate the risk, even adapting some rat data for brain recovery. The idea was to create tools for safer limits. Early attempts, maybe not perfect, but trying to get a handle on it mathematically.
Fascinating. Trying to put numbers on such a complex biological response.
Mm-hmm.
Let's zoom in on the lungs now, pulmonary toxicity. Lorraine Smith identified it, but what are the nitty-gritty details?
Okay, so pulmonary toxicity usually pops up after longer exposures. Think over 24 hours of 100% oxygen at normal pressure, or during hyperbaric treatments between 2 and 3 atmospheres.
And how does it damage the lungs?
One major mechanism, shown in rabbits anyway, involves stimulating something called thromboxane. It constricts blood vessels, promotes inflammation, not good for the lungs. And it's interesting, breathing 100% oxygen for an hour at normal pressure didn't seem to boost free radicals in one animal model, but cranking the pressure up to 2.5 atmospheres did. It suggests the pressure itself adds to the oxidative stress in the lungs.
So it's the combination, again, high concentration and pressure really ramps things up in the lungs.
Seems that way. And if you go even higher, like above 3 atmospheres, HBO can cause really sudden, severe lung injury, especially if you also see signs of CNS toxicity.
suggesting a brain-lung connection?
Yeah, there seems to be one. One theory is that extreme HBO triggers this massive sympathetic nervous system surge from the brain, which then negatively affects the heart's pumping, leading to fluid backing up in the lungs, pulmonary edema.
Wow, so it's not just the oxygen directly hitting the lung tissue, but this whole body system reaction.
Exactly. It can be indirect too. Our chapter also mentions a protein called GDF-15.
Okay.
It's something the body makes under stress, including oxidative stress. And research suggests it might actually be protective during hyperoxia, helping reduce damage. Another protein, P53, seems to be involved in signaling for GDF-15 production when oxygen is high. So the body does try to fight back.
What do the lungs actually look like when this toxicity takes hold? What are the physical changes?
Acutely, you see fluid leaking out edema into the air sacs and the tissue around them. You can get bleeding, protein buildup, then inflammation kicks in.
And if it continues?
If the exposure goes on, you get this proliferative phase. Certain lung cells multiply, fibroblasts lay down collagen. The lungs can heal if you stop the oxygen, but there's a risk of permanent scarring, fibrosis, or even damage that looks like emphysema.
And there's a specific warning mentioned for people with heart failure getting HBO.
Yes, that's important. If someone already has a weakened heart, HBO could potentially make lung fluid buildup worse.
Why is that?
Several possible reasons. It might increase the workload on the heart, cause more oxidative stress in the heart muscle itself, reduce nitric oxide, which normally helps relax blood vessels, make lung blood vessels leakier, or just direct toxic effects on the lung. It really highlights why patient selection and monitoring are so crucial for HBO.
I see. The source mentioned something called a unit pulmonary toxic dose, or UPTD. Was that a way to measure this?
It was an attempt, to try and standardize things, to compare different HBO schedules by relating them back to an equivalent time breathing oxygen at normal pressure.
Did it work?
Not really. It had limitations, especially with how it handled recovery between sessions, and it's not widely used now. Some schedules that look bad based on UPTD don't actually cause problems in practice. And interestingly, divers with years of intermittent high-pressure oxygen exposure often don't show major long-term lung diffusion issues.
That's somewhat reassuring. Lungs might be more resilient to intermittent hits. What about protecting them actively? Any research there?
Definitely. Since free radicals are the main culprits, boosting antioxidant defenses is key. SOD, superoxide, dismutase, and catalase are major natural defenses.
Right.
So researchers are looking at ways to deliver these enzymes directly. Gene therapy is a big one, using harmless viruses to get the genes for SOD and catalase into the lung cells, especially those lining the small airways, may be delivered right down the windpipe.
That sounds pretty advanced. Gene therapy for oxygen toxicity. The chapter also compared normal pressure versus high pressure oxygen effects.
Yeah, a rat study looked at enzymes called nitric oxide synthesis, NOS. They found that the activity of one type, NOS, changed differently depending on the pressure. It went up at normal pressure and also at 2.5 and 3 ATA, but went down at 2 ATA. The other type, NOS, didn't change much. It just shows that the molecular response in the lungs isn't exactly the same when you just increase the oxygen concentration versus increasing the pressure, too.
Okay, let's switch gears from lungs to eyes. Oxygen-induced retinopathy or retrolental fibroplasia. What's the story there?
This is mainly an issue for premature infants. Giving them high concentrations of oxygen, like 100%, can damage the blood vessels that are still developing in their retinas.
Right.
However, the chapter cites one study suggesting that for babies born between 32 and 36 weeks, oxygen therapy over 3 days wasn't linked to a higher risk. And interestingly, another study in premature rats found that HBO 2.8 ATA with 80% oxygen didn't cause this retinopathy, but just breathing 80% oxygen at normal pressure did.
Wait, so the high pressure seemed protective in that case?
It's surprising, isn't it? It suggests maybe the overall hyperbaric environment has a different impact on those developing vessels than just the high oxygen concentration alone. Still needs more looking into, obviously.
Definitely counterintuitive.
Yeah.
Now, our source lists a whole bunch of things that can make oxygen toxicity worse. It's quite a mix.
It really is. You've got other gases like CO2, nitrous oxide, hormones, insulin, thyroid hormones, neurotransmitters like adrenaline, various drugs and chemicals, aspirin, even things like iron and copper.
Wow.
Plus medical conditions like fever, vitamin E deficiency, and even just physiological states like diving, being hot, or exercising. It shows how many things can influence our sensitivity.
Let's unpack a couple. Perfluorocarbons, how do they make it worse?
Right, PFCs. These are liquids that can carry a ton of oxygen, they're being looked at for medical uses, like helping get rid of nitrogen faster after a dive.
Okay.
But because they deliver so much oxygen, especially to the brain, they can lower the threshold for CNS toxicity. Rat studies showed PFCs made seizures happen more easily under high-pressure oxygen.
And trimix diving, that's the helium mix, right?
Yeah, helium, nitrogen, oxygen. Used for deep dives to avoid nitrogen narcosis. But the deeper you go, the higher the partial pressure of that oxygen component becomes. So divers have to be really careful with their mix and dive plan to stay within safe oxygen limits.
Carbon dioxide is on the list, too, and we make that naturally.
Exactly. And even slightly elevated CO2 levels in the body seem to exacerbate oxygen toxicity. The thinking is it might boost that ROS production, maybe even interact directly with some free radicals, makes the oxidative stress worse.
And even just having a fever or working out can increase risk.
Seems so. A higher body temperature ramps up metabolism, meaning tissues use more oxygen, potentially amplifying the toxic effects. Exercise does the same, increases oxygen demand, and demonstrably lowers the seizure threshold. Even high humidity has been flagged in animal studies as potentially worsening things, particularly lung issues and seizures.
It's a long list. It really drives home how context-dependent this is. Okay, let's dive back into the brain, the CNS. We know about the Paul Burt effect, seizures. But what else is going on metabolically?
Well, interestingly, mild HBO, around 2 atmospheres, might actually give cerebral glucose metabolism a little boost without being toxic. And overall brain energy production seems fairly stable up to pretty high pressures, maybe 6 ATA.
Yeah.
But lab studies on cultured rat brain cells show that at those higher pressures, like 6 ATA, you start seeing signs of cell damage, lower survival rates.
Okay.
And significantly, blocking certain things like NMDA receptors or nitric oxide production seem to protect those cells in the lab. That points towards those specific pathways being involved when HBO does become neurotoxic.
So specific molecular pathways get triggered at higher pressures.
Exactly. And while the seizures themselves don't seem directly tied to a big drop in overall energy production, there's often an increase in glucose use right before the electrical signs of toxicity show up on an EEG.
And what about that nitric oxide theory you mentioned earlier?
Right. The idea that maybe prolonged HBO causes too much nitric oxide production. This could potentially override the normal vasoconstriction, the blood vessel tightening that usually happens with high oxygen. If the blood vessels stay too open, you get more blood flow, delivering even more oxygen to the brain, possibly kicking off the toxic cascade.
Nitric oxide seems like a key player, maybe doing different things at different levels. What about other neurotransmitters?
Generally, levels of many neurotransmitters tend to drop under hyperbaric hyperoxia. We talked about blocking NO production being protective in some animal studies, and that this wasn't necessarily linked to GABA levels.
GABA being the inhibitory one.
Right. It also seems like the normal breakdown of norepinephrine and the synthesis of NO kind of shut down during neurotoxicity. This led to the idea that maybe an enzyme called extracellular SOD could worsen things by getting rid of NO too quickly. It's complex.
Definitely sounds like a delicate balance. The chapter mentions studies looking at NO at 2,3 ATA specifically.
Yes. In that range, HBO seems to stimulate neuronal NS, increasing NO. And you can block that increase with NNOS inhibitors or calcium channel blockers. But whether SOD infusion lowers NO seems to depend on the pressure it worked at 3 ATA, but not 2 ATA in one study. And hyperoxia also increases NO bound to hemoglobin. It just shows how pressure-dependent these NO effects are.
Okay, shifting slightly again, ammonia and amino acids, what's their role?
In rats experiencing seizures at 6 ATA, researchers found higher ammonia levels and altered amino acid profiles in the brain.
Why would that happen?
Possibly due to increased breakdown of amino acids, or maybe the glial support cells aren't clearing things properly. The idea is this could create an imbalance between excitatory and inhibitory signals, particularly in the striatum, maybe explaining why seizures can recur if the animal stays under pressure.
So, disrupting the brain's chemical environment. Okay, with all these potential issues, are there early warning signs, biomarkers for CNS toxicity?
Researchers are definitely looking. In animals, changes on the EEG, the brainwave patterns, often show up before convulsions. You see more slow-wave activity than these bursts of abnormal electrical spikes.
So EEG changes could be an early flag, at least in animals.
Potentially, yeah. And even in cell culture studies, you can record changes in electrical activity.
What about the seizures themselves clinically? When do they tend to happen?
Often towards the end of the HBO session or even during decompression coming back to normal pressure. Usually it's a pretty sudden, strong muscle contraction, often with breath holding.
What do you do then?
You have to pause the decompression until the seizure stops, mainly to prevent lung injury from the pressure changes during the seizure. But importantly, having one oxygen-induced seizure doesn't automatically rule out future HBO if needed.
Oh, really?
Yeah, often treatment can continue safely, just maybe at lower pressures or for shorter times. Anti-seizure meds usually aren't required, but some can be effective if needed. And that acupuncture idea, still unproven.
What about people who already have epilepsy? Is HBO a definite no-go?
Well, it's traditionally listed as a relative contraindication, a precaution. The worry is it might trigger a seizure, which could be riskier in the chamber.
Makes sense.
But the source points out that seizures in epileptics getting HBO are actually pretty rare, especially below 2 atmospheres. There's not a lot of recent data, but it does make you wonder if the risk at those commonly used lower pressures has been a bit overstated. The mechanisms might be different from seizures in, say, stroke patients, where HBO sometimes even improves the EEG.
So maybe not as dangerous as once thought, especially at lower pressures.
The consensus seems to be that below 2 ATA, seizures are very uncommon, even if you have a history of epilepsy.
That's a really important practical point. Any other physiological clues? Something called hyperoxic hypernia.
Ah, right. That's basically an increase in breathing rate and depth when you breathe high oxygen. The idea is HBO stimulates the CO2 sensors in the brain.
Okay.
One rat study showed this interesting pattern. A quick bump in breathing and heart rate during pressurization, then later, just minutes before a seizure, another significant increase specifically in breathing rate.
So that breathing spike could be a warning sign.
Potentially, yeah. An early indicator that a seizure might be coming.
Fascinating. Using breathing patterns. Now, what about actual physical damage? Neuropathology.
In rats, you generally don't see actual CNS tissue damage until you get above 4 ATA, and certain things can make it worse. Depressant drugs, high CO2, acetazolamide, ammonia.
And the John Bean effect.
That was the permanent paralysis seen in rats after repeated hits of very high pressure oxygen, like over 5 ATA. At the tissue level, hyperoxia seems to selectively damage the white matter in the spinal cord and brain. Under the microscope, they see different types of nerve cell damage, some cells shrinking and staining darkly, others just breaking down completely.
So real damage at high enough pressures.
Yeah.
Our source has a big table of symptoms for CNS toxicity. It covers a lot.
It does everything from visual stuff like tunnel vision to feeling drowsy or confused, mood changes, clumsiness, tingling, muscle twitches.
Cardiovascular things too.
Yep. Slow heart rate, palpitations, breathing changes, nausea, vomiting, dizziness, and obviously convulsions. Plus general things like looking pale or sweating. It shows how widespread the effects can be.
So given all that, how do you actually monitor for this in a clinic during HBO?
Really, the most important thing is just careful observation by trained staff, watching for any of those signs and symptoms.
What about objective measures?
For the lungs, measuring vital capacity is probably the most practical and reliable thing. It tends to drop before serious damage occurs. EEG, surprisingly, isn't considered reliable for predicting seizures in humans. The changes aren't consistent enough beforehand.
Any other potential markers?
There's some lab research suggesting changes in how red blood cells handle certain fatty acids might be an early sign of cell stress. Also, some studies show blood flow velocity in the brain can increase before symptoms or seizures start at higher pressures, and sometimes dropping the oxygen quickly can stop the seizure.
Is there usually a warning period?
Generally at rest, it seems there's often a delay, maybe around 20 minutes or so, between the first subtle signs and a full seizure. That gives a potential window to intervene.
That window is key. Okay, what about protecting against toxicity? The chapter lists a lot of possibilities.
Yeah, mostly experimental stuff in that table. Antioxidants are obviously a big focus. Vitamin E is used for premature babies who lack it. Though boosting antioxidants with supplements in rats didn't necessarily stop the very first electrical sign of toxicity, it might help recovery between exposures.
Are all antioxidants equally good?
Apparently not. And sometimes what looks good in a test tube, like stopping lipid damage, doesn't actually protect the whole animal. Even lowering body temp hypothermia, its protective effect is debated. HBO can actually cause a slight temp drop in mice sometimes, but it doesn't stop seizures much.
What about more targeted things like that aerosolized SOD?
Right, delivering SOD directly to the lungs. That mice study showed it improved survival and reduced lung edema, suggesting it could be a strategy for severe lung failure from hyperoxia. Other research areas include cytochrome C's role in detoxifying mitochondrial ROS and maybe using drugs like edaravone to counter the increased risk from things like PFCs. And of course, that gene therapy idea for the lungs.
So lots of research, but for clinicians now, what's the main advice?
Number one is just awareness. Know the risks. Even though serious toxicity is rare at typical clinical pressures like 1.5 ATA, monitor patients closely.
Anything else?
Be careful not to assume findings from hyperbaric conditions automatically apply to breathing oxygen at normal pressure. Some things have opposite effects, like desulfurin protecting in HBO, but worsening normobaric toxicity in rats. Even vitamin C, ascorbic acid, might not be purely helpful at high doses. Magnesium has shown some promise in animals' vasodilator, calcium blocker, anti-seizure effect at 6 ATA. Some suggest a magnesium-vitamin E combo, but there's no solid human trial evidence for that yet.
So be aware, be cautious, monitor, and don't assume too much without evidence. Okay, what about developing tolerance? Can the body adapt?
Well, tolerance here mostly means resisting the toxic effects. There might be some adaptation at lower hyperoxia levels, like boosting natural antioxidants, but that's usually not enough for the really high pressures. Drugs face challenges getting where needed, staying effective, not causing side effects.
Well, what about intermittent exposure? Doing HBO in bursts?
That does seem to work, at least in animals. It pushes back the onset of symptoms, maybe allows those protective enzymes like SOD to build up between sessions.
And they try to model this too.
Yeah, more math. Equations to estimate cumulative toxicity and recovery during breaks. They use these to predict latency to EEG changes and found that brain recovery seems to follow a specific pattern over time.
Is that why treatments sometimes include air breaks?
Exactly. Breaking up long HBO sessions with periods breathing normal air is common practice based on this idea, but there is that weird risk of switch-off seizures.
Right, the seizure happening when you reduce the oxygen.
Yeah, it's thought to be the rapid drop in oxygen partial pressure triggering it in susceptible individuals. We still need more research on the best schedules for these breaks.
Okay, one last specific group. Newborn mammals. How do they handle HBO?
They're actually much more resistant to the CNS effects than adults, which is interesting.
But are there downsides?
Potentially. Some indirect evidence in rats suggests HBO might cause prolonged brain vasoconstriction and reduced breathing in newborns. The worry is that exposure could lead to long-term issues, maybe increased tolerance later, but also maybe creating low oxygen conditions in parts of the brain, or messing up how they respond to low oxygen when back in normal air. Could potentially contribute to later neurological problems.
So extra caution needed for newborns. Okay, we've covered a lot of ground. Bringing it all together, what are the main takeaways and the big unanswered questions?
Well, the exact mechanism of CNS toxicity isn't fully pinned down, but the free radical theory, probably involving nitric oxide, is the strongest candidate. The good news is serious CNS problems are rare in standard clinical HBO.
But physicians need to be aware.
Absolutely. And while we lack perfect prevention or treatment, free radical scavengers are used. It's always about weighing benefits versus risks, especially for longer exposures. Maybe someday things like a chemiluminescence index could help gauge individual sensitivity.
What about future directions?
There's interest in maybe using oxygen toxicity therapeutically, like as an antibiotic, or even exploring it for AIDS support, since the virus lacks defenses. We need better ways to measure free radicals in real time to guide therapy. More research into the basic molecular stuff, monitoring multiple body systems at once to define safety limits.
And the tolerance development.
Yeah, studying how cell lines adapt to repeated HBO could give clues. And for the lungs, that gene therapy approach delivering SOD and catalase still looks like the most promising way forward.
So it really hammers home that oxygen, our essential life source, has this complex, potentially dangerous side. We've traced the history, the science, the efforts to manage it. It makes you appreciate the body's balancing act. Any final thought for our listeners?
Just perhaps to consider that intricate balance, how even vital things have limits, and how medicine constantly works to understand and navigate those limits. Oxygen toxicity is a great example, something so fundamental, yet still revealing complexities that require ongoing scientific curiosity to manage safely and use effectively.